Good morning. It's January 8th, and this is your daily brief in science. Here's everything you need to know. Australian researchers have introduced a revolutionary population control method for mosquitoes called the toxic male technique, or TMT. This innovative approach involves genetically engineering male mosquitoes to inject venom proteins into females during mating, aiming to manage mosquito populations without complete eradication. The significance of this method lies in its recognition of mosquitoes as vital pollinators and food sources within ecosystems. The TMT specifically targets female mosquitoes, significantly reducing their lifespan and thereby limiting the transmission of diseases such as malaria and dengue fever, which affect millions globally each year. Laboratory tests indicate that females mating with TMT males experienced a lifespan reduction of 37 to 64%, with blood feeding rates decreasing by 40 to 60% compared to traditional control methods. The venom proteins utilized in this technique are sourced from the banana spider and the Mediterranean snakelock's sea anemone. These proteins are toxic when injected but less harmful when ingested, ensuring that non-target species remain safe. Unlike conventional pesticides, which can adversely affect beneficial insects like butterflies and bees, the genetically modified mosquitoes are designed to spare these vital species. The TMT is projected to be as effective as existing pesticides while addressing the growing challenge of pesticide resistance. However, it is important to note that this technology is still in its early development stages, with field trials expected to begin in 5 to 10 years due to regulatory and safety requirements. The research findings were published in the peer-reviewed journal Nature Communications on January 6, 2025, establishing a proof of concept for the TMT. This project is a collaboration with UK company Aussie, focusing on managing invasive mosquito populations by promoting the mating of modified males with females. Genetic engineering has historically been employed to control disease-spreading mosquito populations, primarily through the release of sterile males. However, this new method directly addresses female mosquitoes. While the timeline for releasing these genetically modified mosquitoes remains uncertain, researchers are actively engaging with local communities in areas impacted by mosquito-borne diseases. A recent study led by Dr. Lu Qi from Tulane University has found that the timing of coffee consumption plays a significant role in health outcomes, particularly regarding cardiovascular disease and overall mortality. Researchers analyzed data from over 40,000 adults aged 18 and older, collected from the National Health and Nutrition Examination Survey conducted between 1999 and 2018. Participants categorized their coffee intake into morning, afternoon, and evening consumption. The study revealed that those who drink coffee in the morning have a lower risk of cardiovascular disease compared to those who consume it later in the day or not at all. Interestingly, even individuals who consumed 1 cup or less of coffee still showed a reduced risk, although this effect was less pronounced than for those who drank more. The follow-up period of the study lasted nearly a decade, during which 4,295 deaths were recorded, including significant numbers attributed to cardiovascular disease and cancer. Dr. Qi emphasized that dietary guidelines may need to incorporate timing advice regarding coffee consumption, suggesting that when coffee is consumed could be as important as how much. This study highlights the importance of timing in dietary recommendations, indicating that future guidance may benefit from considering such factors. While the study establishes an association between coffee timing and health outcomes, it does not prove causation, and its observational nature allows for potential confounding factors. The study controlled for various health factors like diabetes and hypertension, yet further research is necessary to confirm these findings across different populations. Experts also note that sleep quality plays a crucial role in heart health, with poor sleep potentially increasing the risk of heart disease. Engineers at the University of Pennsylvania have made a significant advancement in quantum sensing with a new method that enhances nuclear quadrupolar resonance spectroscopy. This technique enables the detection of signals from individual atoms, a breakthrough that could change the landscape of drug development and protein research. Traditionally, spectroscopy methods average signals from trillions of atoms, making it difficult to detect subtle variations between individual molecules. This has been a critical limitation in fields like protein research, where minute differences can have substantial implications. Lee Bassett, the senior author of the study, highlights that this new method reveals previously hidden details about molecular structure and dynamics at an unprecedented scale. The precision of this technique allows researchers to gain insights into molecular interactions at the atomic level. This capability could significantly enhance drug development processes by uncovering molecular phenomena that were previously concealed. The discovery was sparked by unexpected patterns observed by Alex Breitweiser during experiments with nitrogen vacancy centers in diamonds, which drew on historical nuclear magnetic resonance studies. Collaboration with researchers from Delft University of Technology helped refine the method, allowing for the accurate capture of single atomic signals. The study received backing from the National Science Foundation and included contributions from various researchers, such as Zhu-Yung Huang and Tim H. Taminow. The advancements in technology have enabled the measurement of effects that were previously unattainable, marking a notable breakthrough in quantum physics and spectroscopy. This new method's ability to isolate individual atomic nuclei is akin to extracting individual data points from a vast dataset, providing deeper insights into molecular properties. As researchers continue to explore this technique, it holds promise for addressing significant scientific challenges in the future. A breakthrough in kidney injury detection has emerged, with findings published on January 6, 2025, in the journal Clinical Pharmacology and Therapeutics. Led by Dr. Sushruth Waikar, the research team has identified 6 new biomarkers that promise quicker and more sensitive detection of kidney injuries, a development that could enhance drug safety and patient health outcomes. These biomarkers are primarily produced by the kidneys in response to injury or inflammation, allowing for a faster diagnosis compared to traditional methods like serum creatinine blood tests. Early detection of kidney damage through these innovative biomarkers enables prompt clinical intervention, which may help mitigate long-term damage and improve patient outcomes. Drug-induced kidney injury, known as nephrotoxicity, is a common complication in clinical settings, often arising from medications such as anti-inflammatory, antibacterial, antiretroviral, and chemotherapeutic agents. This condition can lead to treatment discontinuation or restrictions, significantly limiting patient options. In this study, the research team measured urinary levels of these protein biomarkers in both healthy volunteers and patients undergoing chemotherapy for mesothelioma, a drug recognized for its nephrotoxic effects. The team intends to explore the broader application of these biomarkers for monitoring kidney health in clinical trials. Dr. Waikar also serves as a consultant on nephrotoxicity biomarkers for several companies, including Vertex, PepGen, and Ikena. Notably, while current biomarkers are often too slow to indicate early kidney injury, the newly identified biomarkers can be detected in urine and may reveal damage within a 24-hour timeframe. A recent study involving 590 pregnant women in Malawi has unveiled promising results regarding the treatment of anemia. The trial demonstrated that a single iron infusion during the 3rd trimester significantly reduces anemia compared to traditional oral iron tablets. Specifically, only 46.7% of women in the infusion group were affected by anemia at delivery, in stark contrast to 62.7% in the tablet group. This research, co-led by the Walter and Eliza Hall Institute in Australia and the Training Research Unit of Excellence in Malawi, marks the first time that iron infusions have been shown to effectively enhance iron levels in pregnant women approaching delivery. Professor Santarém Pasricha highlighted the global importance of addressing anemia during pregnancy, emphasizing that iron infusions are superior to daily tablets. Currently, the World Health Organization recommends oral iron tablets as the primary treatment for maternal anemia. However, the findings from this trial are prompting researchers to advocate for updates to these guidelines. The implications of this research could potentially transform maternal care practices worldwide, including in countries like Australia, ultimately improving birth outcomes. Adequate iron intake during pregnancy is crucial to prevent complications such as preterm delivery, low birth weight, and postpartum depression. Anemia during pregnancy poses significant risks to the developing baby, potentially affecting placenta development and brain growth. Globally, iron deficiency is a leading cause of anemia in pregnancy, impacting approximately 37% of pregnant women, which equates to nearly 32 million women. In Australia, around 25% of pregnancies are reported to involve anemia, increasing the risk of complications for both mother and baby. The success of the trial indicates that intravenous iron can be safely administered even in basic health centers in remote areas, thereby broadening access to effective treatment. Moreover, the sustained impact of iron infusions on postpartum anemia levels is an unprecedented finding that supports their use in late pregnancy. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.